Hello everybody and welcome to biomolecules again. So, we are discussing module 5 on the biosynthesis of proteins, how proteins are synthesized in our biological cells, and we have seen that it goes through two distinct processes or two distinct steps. The first one being the transcription, where we have seen how the information present in the nucleus comes out of the nucleus with the help of mRNA. So, this is the process of transcription was about synthesis of mRNA, messenger RNA, from the double stranded DNA that was present in the nucleus of the cell. And here we have seen that the sequence of the mRNA is exactly the same as that of the antisense strand of the double stranded DNA. So, very quickly, if you have the 5 prime to 3 prime DNA and the 3 prime to 5 prime, this is your DNA that is present in the nucleus, and then so this was called the sense strand, and this one was antisense strand, and then we have seen that the antisense strand was taken as the template to synthesize the new mRNA. With the help of the mRNA polymerase. So, therefore, that is how the sequence of the mRNA would be complementary to the antisense strand, and therefore, the sequence of the newly synthesized mRNA would be exactly the same as that of the sense strand. So, that is how all the genetic information that was present in the sense strand has been transcribed or transcripted into the newly synthesized mRNA. And then finally, of course, the mRNA, after the process of splicing, it undergoes maturation to the matured mRNA by removing all the unnecessary parts of the mRNA, taking only the important part of the mRNA for the synthesis of protein. So, basically which means the coded part of the mRNA would be kept, and the rest of the parts which do not code for proteins or which do not code for amino acids would be removed. So, that is how you get a matured and functional mRNA that is fit to be used for the synthesis of the proteins, and this is outside the nucleus of the cell in the cytoplasm. So, that was the first step about transcription, and then we have talked about the second step, that was translation. So, in translation, basically the mRNA that you have synthesized from the previous step would be used, all the information present in the mRNA would be used to synthesize proteins, or I will write polypeptides. So, mRNA to polypeptides is the process of translation, and we have also discussed why this is called translation, because it changes from one language to the other. The language of nucleic acids is being changed to the language of the amino acids. That is why this is called the translation process. And in the last lecture we have discussed that a fair number of biomolecules are involved in this process of translation. We have talked about, of course, this present mRNA, that is one of the key ingredient that would be used. We have also seen how tRNA was involved, and we have discussed a detailed about the tRNAs. So, this is the structure of a typical structure of a tRNA, which is basically a single stranded RNA molecule, starting with the 5 prime end and completed at the 3 prime end with a free hydroxyl group. So, it is a single stranded RNA which has self hybridization in some regions of it. This is one, this is one, this is one, this is one, self complementary sequences present here. And we have seen that there are some modifications here and there present. Those modifications were important to give the flexible structure wherever necessary in the tRNA. Two important parts in the tRNA which would be absolutely essential in determining the amino acid sequences or in synthesizing the polypeptide chains are, one is this, that every tRNA has a 3-letter code, nucleic acid code. This is RNA code, 3 nucleobases long, which is known as anticodon. This is very essential for us, and the end of it, 3 prime end of it, has a free hydroxyl group, that is also very essential for us. And always the tail, or known as the acceptor arm, has a sequence of CCA. This is basically a 7-mer sequence, and the last 3 are CCA. Adenine is the last nucleobase present in every tRNA with a free hydroxyl group at the 3 prime end, and that 3 prime end actually reacts with amino acid to produce your monomer. So, tRNA here, and we have seen in the last lecture how ATP was involved in this case. So, I am writing tRNA and OH. OH is the free hydroxyl group on the adenine, and here there is the anticodon of it. Anticodon is there. Now, if you have an amino acid, let us say it is R, NH2 and the carboxylic acid, with the help of ATP, this forms or this is your NH2. So, there is amine group, there is an acyl group. So, this is called amino acyl tRNA. This is amino acid which is conjugated to tRNA via a acyl bond. So, this is called amino acyl tRNA, and this reaction is catalyzed by amino acyl tRNA synthetase. This is the enzyme that catalyzes this reaction. This enzyme is also known as tRNA ligase. tRNA ligase. It basically ligates the carboxylic end with the 3 prime free hydroxyl end of the tRNA. So, ideally every tRNA carries a specific amino acid into its 3 prime end. So, this is the 3 prime end, and every tRNA has an anticodon associated with it. So, every amino acid actually now has an code attached to it which we call as anticodon. So, these two are the ingredients we have talked last lecture, and today we will talk about the other part of it, another very important ingredient or another very important class of biomolecules is a ribosome. Or ribosomes. Ribosomes are absolutely essential for the process of translation. It is basically a combination of proteins and RNA known as rRNA. rRNA means ribosomal RNA. So, ribosomes is basically an assembly of proteins, multiple number of proteins or many units of proteins are there, plus some RNAs called rRNA, ribosomal RNA here. So, ribosomes or ribosomal RNA has or are of globular shape. It triggers the synthesis of the ribosomes. So, ribosomes are originally synthesized from rRNA, and in the assembly both are present, ribosome subunits are present, at the same time rRNA also are present, and this is very important. What does it do for the protein synthesis? It actually provides a workbench or a vessel or a container. It acts as a container on which the synthesis of protein occurs. It acts as a workbench for protein synthesis. rRNA and proteins make up the large and small subunits of ribosomes. I will show you the structure of ribosomes. Ribosomes are the site of translation where the polypeptides are actually synthesized. So, these are the key people or the most important people who have worked pretty much on the ribosomes to find out the structures of ribosomes, plus to find out how the mechanism works, how the polypeptides are synthesized on the ribosomes. Of course, ribosomes were known much beforehand, and there are many other properties, and ribosomes were that time regarded as that it is very important for the synthesis of proteins. tRNA was also regarded as very important for the synthesis of proteins, but actually how the mechanism works, how the amino acids are coming close to each other, what is the proper function of ribosomes, all these things were developed by these three people who have received Nobel Prize in Chemistry in 2009, very recently actually. She is Professor Ada Yonat from Israel. He is professor, of course, I think you can recognize him, Venkatraman Ramakrishnan. He is also popularly known as Venki Ramakrishnan. He is in the from the Cambridge University, and Professor Thomas Seitz from USA. So, these three people have worked to find out exactly the mechanism of the functions of the ribosomes, and also they are the pioneers of showing how exactly antibiotics works on the ribosomes. So, what is the function of ribosome? What does it do? It actually works as I have mentioned, it works as a workbench, so workplace, or in other words it works as a container where all the other ingredients comes and will be accumulated in this place, which means once you have a ribosome, on the ribosome mRNA will come and bind. And then tRNA will come and bind. So, that is one tRNA. There are many tRNAs that are present in the cells, each carrying a specific amino acid and each carrying a specific codon, anticodon. If that anticodon is complementary to the codon of the mRNA, then that specific tRNA, I write AA for amino acid or amino acyl. That particular tRNA which has a matching or complementary anticodon to this will come and bind here. So, naturally that means that specific amino acid will also come and bind here. Similarly, the other tRNA also has to come because you have to synthesize a peptide bond. So, you need 2 amino acids. So, ribosome provides that space on which you can have at least 2 numbers, actually 3, close to 3 number of tRNAs to come and sit down here side by side, so that they can undergo reactions. And of course, mRNA is providing as the template where the tRNA will come or which tRNA will come, that specificity comes from the mRNA. The workbench or the space is given by the ribosome. So, this is a structure, typical structure of one ribosome. This has 2 units called 50S subunit and another one is 30S subunit. 50S ribosomal subunit and 30S ribosomal subunit. And each ribosome has, as I was talking about space for 1 tRNA, for 2 tRNA, for 3rd tRNA. So, they are called sites in the ribosome. So, there are specific parts or spaces inside the ribosomes where the tRNAs can come and bind. So, one of these is called the A site. So, this is A site where a particular tRNA will come and bind. There is P site here, this P site is also a space where another tRNA will come and bind, and then there is a third one called the E site where there is a little bit of space for another tRNA. So, each ribosome contains 3 sites: A site, P site and E site. A site is actually known as amino acyl site. P site is where the peptide synthesis occurs. So, this is called peptidyl sites. Peptidyl site. And E site is where it knocks out or exit site. It after the synthesis it goes off. The tRNA goes off actually. We will see. E site is called exit site. And I have given different names for this just to make it easier to remember. A site, I call them approach site, because here the first tRNA is coming or the new tRNA is coming, not the first, the new tRNA is coming, approach site containing the amino acid. P site is where the reaction is actually taking place, and I call it present site, which is always there at the middle of it, present site. And E site is exit site where it is going out. So, this picture I have taken from the Nobel lecture of I think Professor Ada Ionut or Professor Venki Ramakrishnan. So, the entire 70S ribosome with mRNA and tRNA. So, this is the picture drawn from a structure at 2.5 angstrom. This is the resolution. The short section of mRNA in this structure is barely visible because it is hidden somewhere here in magenta, because it is buried in a cleft in the 30S ribosomal subunit. It is buried here actually. So, the reason Professor Ada Ionut, Professor Venki Ramakrishnan and Professor Stites have received so much fame, because that they have done something really beautiful. What they have done, they have crystallizes, they have obtained the crystal structures of each step when the protein synthesis was occurring. Ribosome with tRNAs, how do they look like at each step? So, it is a huge work of course, and really a new kind of idea of crystallization, because they had to improve the method of crystallization. They had to invent new equipment, new instrument to crystallize them in the frozen form and in real time. So, they basically have found out each real structure. You can really see the structure in each step, how they are working. So, this is another picture of the ribosomal subunits 30S and 50S here. This has been taken from the Nobel lecture of Professor Adayonat, and you can see this is the E site, this is the P site, and this is the A site. Same is here. It's the A site, P site, and the E site. So, this shows the crystal structures, how the crystal property have been improved over time. So, 1980s, you can see there's these are the data that were available that time, barely readable. 1984, you can see there is fairly nice crystals, single crystals that they could achieve. And then 1990, of course, they have this very high resolution crystal structures that have been obtained. This is a pure crystal structure of a 30S ribosomal subunit, showing this really in the single crystal forms. All these crystals have been obtained in cryo form, cryocrystals they call it. Okay. So, now comes how the synthesis works, how the mRNA is used, how the ribosome is used, how the tRNAs are used to synthesize the peptide bonds. And this occurs in a sequential steps. The first step, so we are now discussing about the mechanism of translation. The first step is called the initiation. Initiation is the step where the first tRNA comes and assembles over the ribosome and also the mRNA. So, if you have the ribosome here, the mRNA comes and binds, all the ribosomal subunits comes and binds here. So, it is basically an aggregation of the proteins as well as the mRNA that makes a ribosome. Once mRNA comes and gets into the, it docks into the protein structure or the aggregation structures and gets stabilized there, containing the codons, the genetic informations. Initiation is where the first tRNA would come and bind. So, I am not drawing the whole thing, I will only draw the last one. Here this is the codon, anticodon part which is important, and this is what is important. OCO, CH, let us call it R1 and amine NH2, the first amino acid attached to the tRNA, and here is of course your adenine. So, this is all happening at the 3' end of adenine. So, the first tRNA comes and bind to the mRNA sequence, and of course, when the tRNA comes, it has to have the anticodon which matches or which is complementary to this sequence. It happens that the first amino acid is always a methionine, or we call it met, and it has to start somewhere. The synthesis or process of initiation, the binding of the first tRNA on the mRNA always happens with a start codon. So, mRNA, wherever it has a sequence of A, U, G, this sequence is called the start codon. So, mRNA has to have this sequence somewhere where the ribosome has bound or all the other ingredients have been bound, and this sequence will be the starting point. AUG will be the starting point. So, the tRNA should have the anticodon which is complementary to AUG, which basically is TAC, not TU, uracil, I am sorry, UAC would be the anticodon of the tRNA, and this anticodon is always attached to this amino acid methionine. So, as I have mentioned that each tRNA, this is again and again I am repeating and I am writing now, because this is absolutely important. Each tRNA has a specific anticodon and carries a specific amino acid. At the 3' end. So, that is the identity of a tRNA. It should have a specific anticodon and it should bear a distinct amino acid, that is all. So, if the anticodon is complementary to the codon of the mRNA, then they will hybridize, and this hybridization would be further stabilized by this assembly. So, that is the process of initiation. First tRNA carrying the first amino acid, which is methionine, comes and binds at the start codon of the mRNA, which is AUG. Now, what next you need? You need to bring another tRNA, so that there will be two amino acids which may react. So, the second step is known as elongation. Elongation is the step where the peptide synthesis occurs. In this step, peptide synthesis occurs. So, basically here you need one tRNA, two tRNA, this is AUG. So, that is the starting point, and this tRNA carries one amino acid methionine, and this tRNA carries one amino acid, I am writing AA. So, these two now are coming close in proximity, and now they can react to produce a peptide bond, and then it will go on and on subsequently. So, you get, that is why it is called elongation, the peptide number of peptide bonds is increased. So, you get a polypeptide. Polypeptide or I called polypeptide bonds. That is the step of elongation here. So, I am now the translation overall, then I will show you the chemical mechanism. The process by which amino acids are synthesized from mRNA with the help of ribosome and rRNA, that is called the translation. The information in mRNA is decoded to synthesize polypeptides, that is what we have been talking about. So, the whole process is broadly divided into 3 steps. First step is initiation. The ribosome assembles around the target mRNA, as I have shown here. This is your mRNA, ribosome would come, or in other word, the mRNA goes in, find out the ribosome and stacks there. So, one important thing should be very well clarified, that ribosome works as I have said, as the workbench or the vessel, which means ribosome is static. This is very important. Ribosome is static, it does not move. Ribosome stays in one place. All the other ingredients, they move if need be. mRNA would move, tRNA would move, but ribosome would stay intact because it is a workbench. So, bench does not move. So, initiation is where the ribosome assemblies around the target mRNA. The first tRNA then brings the first amino acid, which is always a methionine, to the start codon and initiate the peptide synthesis. Then comes the elongation process. Elongation is the tRNA then continues transferring amino acids one by one to the previously attached amino acids, which corresponds to the next codon of the template mRNA. I will explain. The ribosome then moves to the next mRNA. This is mistake. Ribosome does not move. Sorry. Ribosome does not move. mRNA moves to bring the new tRNA or to accommodate the new tRNA. So, here. So, this is your tRNA. You have ribosome, you have the mRNA. And then, so this is A site, this is P site. So, first tRNA will come here, tRNA 1, that is the first tRNA. Here comes the tRNA 2. Now, these two will react, produce a peptide bond, and then once this peptide bond is synthesized, one of them would be knocked out because one of them will lose the amino acid. I will show you. And then the third tRNA, which would be complementary to this codon, has to come, tRNA 3, but this is outside the ribosome. So, it has to get into the ribosome, then only you can have the good hybridization. So, what is done is mRNA, after this is the work of this first tRNA is finished, it pulls the mRNA here, so that in the next phase this codon is out of the ribosome, this is still there at this end, and this codon comes very much into the workbench or into the vessel, and that is how the new tRNA would be accommodated there, and that is how the process would go on. Once a peptide is synthesized, mRNA would be pulled, will bring the new codon into the ribosomal subunit part or into the assembly, and then it will go on and on. The third one is the last one, the termination. Termination is the process, is the termination of translation actually. When the polypeptide chain is synthesized, peptidyl transferase tRNA. So, the enzyme that catalyzes the synthesis of the peptide bond is called the peptidyl transferase tRNA, that encounters a stop codon. So, when it reaches the stop codon of the mRNA, then the protein synthesis is stopped, because the stop codon of the mRNA, that is complementary to an anticodon of a tRNA, and that tRNA does not carry any amino acid, is devoid of amino acid. So, the complementary sequence of the stop codon does not carry any amino acid. So, no further protein synthesis can occur, that is where the synthesis will stop. The polypeptide chain then detaches from the mRNA and the ribosome also dissociates, which frees the mRNA also. So, the polypeptide chain gets folded. So, you have synthesized a long chain of peptide with many amino acids, and of course, you have seen before that how internal interactions are present within the amino acids that will be responsible or that forces the structure to fold itself, right. So, immediately whenever the polypeptide chains are synthesized, it will be folded to produce a functional protein. Sometimes what happens, the folding also starts during the synthesis itself. So, even before the termination occurs. So, sometimes the folding as well as the translation occurs at the same time. So, now I will show you the chemical reaction, but before that here this will clarify better. This is called E site or exit site. This is P site, peptidyl site, and this is A site, amino acyl site. I call it approach. This is where the new tRNA is coming in. So, it starts with this here, the last, this is the end of it and this is the start of it. Peptidyl site, or you can call it present site where things are happening, and this is exit site. Okay. So, here in this example it has been considered that you have already synthesized a peptide bond and you are trying to synthesize n plus 1, n number of peptide bonds have been synthesized and you are trying to bring in the n plus 1 amino acid. So, somewhere in between we are discussing. So, tRNA CCA 3 prime that is connected to a new amino acid, and this has been a peptide. So, this is the nth number of amino acid, which means you already have a polypeptide chain there. This is the E site, which is existing exit site, empty for the time being, and this is the new tRNA or the new amino acid attached to the new tRNA that is present in the A site. What happens is these two will react. I will show you the reaction. These two will react, and once it reacts, after that this bond, this whole thing would be transferred here. As a result, this RNA, this tRNA here would be devoid of the amino acid. This does not have any amino acid now, because that amino acid has been reacted here and it has become a free tRNA without the amino acid with the free hydroxyl group. Now, since this does not carry an amino acid anymore, it has to go out, because that is how the new tRNA again will come. So, the new tRNA comes, knocks it, punches it, that eliminates the tRNA which was there to the E site. So, P site after the synthesis goes to the E, approach side goes to the P. So, that is how it happens. Every time a new tRNA comes and every time a polypeptide is synthesized, this will be knocked, this mRNA would be pulled to accommodate the new tRNA which has the codon here. So, this should be pulled, this part will get into the ribosome, this new tRNA will come in and the old tRNA would exit. And always we have to remember that ribosomes act as the static workbenches on which peptide synthesis occurs. The mRNA moves along the ribosome, as I was saying that mRNA moves, once the synthesis is done, mRNA pulls itself. Once a mRNA codon binds to the correct tRNA attached to a specific amino acid. Now, let us look at the reaction, how it happens, and then I will show you a real video where you can see how this is happening really. So, we have tRNA, phosphate, Here is your adenine. So, here is your adenine, this is the 3' end where the amino acid is bound, and we will take that already one peptide bond has been synthesized. Let us say this is R1, NH, CO, R2. I will do it other way around. This should be R2, this should be R1, and here you have the terminal one. So, we have taken the example that already one peptide bond has been synthesized and a third amino acid is coming into the picture. So, this is the approach site. Adenine, OH, this is H, O, and here the new amino acid is present. Let us say this is R3 and with NH2 or NH3 plus, whatever it is. The reaction is the free amine group present in the A site, this is A site, this is P site, that will react to the acyl bond, eliminating this. So, cleavage of the ester bond and formation of an amide bond. So, if this happens, then what do you get? You see, you get a cleavage of the P site. In the P site, what you will have? You will have a free hydroxyl group on the adenine without any amino acid, and this whole of this peptide chain would be transferred or would be conjugated to the new site. So, what you see, what you get actually is tRNA, O, A for adenine, here also the free hydroxyl group plus the actual one, O minus, O, CH2, adenine, OH, H, O. There would be 3 amino acids here. The new one would be there first, because yes. So, the other one has been attached after the 3. So, this should be 3 NH, CO, CH, R2, NH, CO, CH, R1, and the last one is amine. So, all 3 amino acids should be attached here, A site, this is your P site. So, you see the job of P site is done, job of this tRNA is done, it does not contain any longer any amino acid. So, it has to move, it has to be relieved. On the other hand, A site become your major one. So, what will happen? tRNA that contains, I am not writing all the other part of it, let us say CHR4 amine. This is the new tRNA. So, this will come, and this of course, the anticodon has to get hybridized to the mRNA. So, the mRNA would be pulled, this would come, this would push, you have limited space there, this would push and this would knock this. So, the tRNA will be exit, which was originally present in this site, P site would exit, this comes will take the place of P site, and this will be your new A site. So, that is how the reaction goes. Now, let us look, I have a video here which actually models. This video has been created by Professor Ada Ionat, and she has actually visited our IIT few years before and gave a lecture. So, she has shown this video. I have copied this video from YouTube from her site, and this is the link. You can find the video in this link, which actually shows from their crystal structures what they have obtained. Hopefully, it will be visible. I have to show it couple of times, I guess. So, this is called ribosomes in action. So, this video has been done based on the crystallographic studies, and of course, it is already mentioned here, Ionat's group, the Weizmann Institute, Israel, and Max Planck Research Institute, Hamburg, Germany. So, here you go. I have to pause a time to time to explain it. So, this is the mRNA sequence that is getting, and this is your ribosome. So, mRNA is going to get into the ribosome. mRNA carrying the genetic code approaches the small ribosomal subunit. And it is getting into the interior initiator tRNA charged with an amino acid is brought by IF2 to the P site. Inter subunit bridge are formed. A little bit back, I think I have to show. So, amino acid tRNA approaches from the left, assisted by L7 and stalk, it reaches the A site. Here. So, this is the exit site. This is the structure of the tRNA, and you will see, if you look here as it will proceed, there will be a small fluorescent blue bright fluorescent color on the tip. That means that is the amino acid that is attached to the tRNA. This is tRNA. Here on the tip you can see something is glowing as it will move, that is the amino acid. So, here it is not present because the amino acid has been taken away here, and this is only the 3' hydroxyl end. So, it will exit, knock out. Here again this is another tRNA that is getting out. So, all tRNAs are getting out. Here you can see this is the amino acid. This is the new amino acid, new tRNA which is approaching. So, this is approach site when the new tRNA is getting into the ribosome. This is the amino acid. Here you can see 3 of them. So, this is exit, this is P site, this is approach site. So, forget about this now. This is exit site where the job of tRNA has been done, free hydroxyl group. So, this will leave. This is the P site where you have already synthesized a good number of peptides, the present site, and this is the A site, approach site, which is carrying the new amino acid. So, these two will react, whole thing would be transferred here, this would become free and it will eliminate. So, it is knocking here, this everything is transferred and it becomes like this. And all the time the mRNA is moving itself in this direction. Now, the nascent protein, which means the just right after the synthesis, emerge from the exit tunnel opening from the exit site. This is the protein. And it reaches the stop codon. Stop codon reached the decoding site. So, again stop codon means it is the code, the complementary of which is the anticodon of the tRNA, and that particular tRNA does not contain any amino acid. So, it will be stopped there. Ready for next act. So, these are catalyst basically. So, end of the process, the two subunits of the ribosome, they dissociate themselves, ready for the next act. A new set of mRNA will come, or mRNA is already present, a new set of tRNA will come and the new protein should be synthesized with the same ribosome. So, I will play this video again. Here you can see, you can find out by yourself. I will not talk now. Okay. So, this is how the mechanism works, and of course, I have once again, if I go back to the reaction, you can understand how it was being transferred to the new one. The new was working here. This A site was taking all the other peptide that was already synthesized in the P site, that was converted here, and we were finally getting everything, and this A site was moving to the P site was moving to the exit site, and the new tRNA was taking the place of A site, and the elongation goes on. So, that is the chemistry behind it, and of course, this is catalyzed by the protein, which is as I mentioned, is the transferase. All right. So, with this I will conclude today. Still a little bit of content is left. on how the codons are present or how the codons came into the effect, and some more calculations we can do, and that we will do in the next lecture. Thank you.